Hello and welcome back to microbiology. Today we're going to do chapter 10, which is about genetic engineering and recombinant DNA technology. This is a unique chapter for us because this is going to talk about a lot of advanced lab procedures. Overall, this stuff is super cool, but not a lot that I'm going to test you on. So as a general idea, I'm going to test you on the things that we'll do both in lab and in lecture. So things like PCR, topics like gel electrophoresis, or even cloning, recombinant DNA technologies. Those are what would be more testable for us. This will get to a more ethical discussion at the end, so please again keep that in reference whenever we're thinking about what's going to be on the test. So what we talk a lot about in lecture is the theory behind the science, but we do much less on the application. And really, theory is great, except for it doesn't serve us much good unless we apply it in the laboratory. So this chapter is important to understand that application is really the bigger outcome of what our sciences do for us. So naturally, whenever we go through this, try to take normal theory and think about it in a new abstract way. And that's the way we actually get these advances that lead to eventually even more theories. So this idea that research and theory go together, that without one we couldn't have the other. So let's even start with something simple like the intrinsic properties of DNA. What naturally happens with DNA? Well, if you have two opposite strands or complementary strands, they'll want to stick together naturally due to hydrogen bonds. But what do you think heat does to those hydrogen bonds? Well, it breaks them. But if I throw separate or single-stranded DNA in with its complement, whenever we cool it back down, it should stick together. So naturally, we have some potential uses in things like gene probes or hybrid. Restriction enzymes or endonucleases are another interesting topic. Again, all they naturally do in the system is cut DNA that they recognize. So they're looking for typically palindromic sequences that they know. And so, again, in nature, they're there to cut up DNA that doesn't belong. But we could use them in unique ways, like the ability to form sticky ends. Again, what a restriction enzyme or endonuclease looks for is a palindrome. Palindrome is going to be the same forwards as backwards. And as a result, every time that an endonuclease or restriction enzyme recognizes a palindrome, it's going to cut it. But sometimes it cuts it in such a way that we get a staggered end with some overlap that allows for some initial kind of hydrogen bonds to form, holding DNA in place. Let's look at that now. As you can see here, what restriction enzymes do is look for very specific sequences, like with EcoR1. EcoR1 is simply looking for the sequence GAA-TTC. And every time it sees that sequence, it's going to cut between the G and the first A. So again, palindrome, same forwards as backwards, right? So what we get is this staggered in. But if I cut the DNA of the old organism and of the new organism with the same restriction enzyme, we get this overlapping feature that allows us to insert new DNA into pre-existing DNA quite easily. But that's again requiring that we use the staggered cuts. But what about something like HAE3? HAE3 is going to make a blunt cut. Notice we're looking for GGCC here. And so we'd cut straight across, which results in a blunt. That blunt cut doesn't use for this kind of insertion because there's no overlap or hanging that allows for that base pair matching. And so really, things like HAE are used for other applications. A good other application is restriction fragments. Restriction fragments are chunks of DNA created whenever we cut the DNA up. But we can actually use those chunks and gel electrophoresis to produce what we call a restriction fragment length polymorphism. The idea is pretty simple. If we cut the DNA of an organism with a restriction enzyme and then cut the unknown DNA with the same restriction enzyme and we see the same size chunks, that suggests that that is in fact the same DNA. So we can use RFLPs to actually do things like identify things. That's why, again, it's even usable in the court of law. Ligase is another enzyme that had specific application. In the case of the real world, it was used to stick DNA to DNA. That's not different here, but do recall that if we move genes from one organism to another, they're just going to be loosely held in through those sticky ends. But if we apply ligase, now we permanently lock the DNA in. Another interesting enzyme is reverse transcriptase, which simply just turns RNA into DNA. And HIV does this to again allow for the insertion of the viral RNA genome into the host DNA. And so there are interesting applications for these things. For example, if we tried to plug eukaryotic DNA into prokaryotes, it wouldn't work because eukaryotes have essentially non-coding regions that were, again, not used to make proteins. And so we'd have to have those removed before we can plug the DNA into a prokaryote. So let's say I want to put the human insulin genes in bacteria. Before I could do that, I must take and remove all the regions of non-coding, which we called introns. Good news is this naturally happens. As we make the precursor mRNA, we then modify it through splicing out of, again, introns and produce a messenger RNA product that is ready to be turned into a protein. But if I use reverse transcriptase, I can turn that messenger RNA product into DNA and then copy the corresponding strand of DNA to produce what we call double-stranded cDNA or complementary DNA. The idea is now that cDNA can be plugged into a prokaryote and we would make exactly the protein that was needed to be made. Another cool thing we can do is something called gel electrophoresis. And really all gel electrophoresis does is push DNA based off of charge and size a specific distance. And so the idea is DNA is typically negatively charged, and as a result, if we run it through a gel with a negative end at one pole and a positive end at the other, the DNA would move away from the negative end towards the positive pole. But since the gel is a matrix, different sizes of DNA will move different speeds or rates. And as a result, based off of size, we can really determine how big or how far the DNA should move in the gel. And so the idea is if I cut the same DNA with the same restriction enzyme, it should consistently give me the same banding pattern. And again, that's how gel electrophoresis and RFLPs really work. And so that's what we see here. Notice we use the same restriction enzyme to cut the DNA to produce different size bands. Okay, and based off of, again, size and the charge, we will move different distances, and so we can use that as comparison to, again, relate DNA sequences we think are the same or similar. Nucleic acid hybridization and probes are something I'm going to be less likely to test you on. It's not that they're not important, it's just it's a newer topic. So I want to point out how these things would work though. If I had a single strand of DNA, but also its single strand complement, if the temperature was cold enough, they would want to stick to one another. So the idea if it's not above denaturation point. And so the idea is I can actually test for identity. And so if I have this gene that I know is only found in E. coli, and I throw it in a warmed mixture of unknown DNA, but whenever I cool that mixture, it sticks to the unknown DNA, what does it tell me? Well, it tells me that that must be E. coli, because that gene that I plugged in was only found in E. coli, and as a result, I can now establish identity. Naturally, we do need mechanisms for recognition of whether the DNA stayed, so enzyme-linked probes or even fluorescent reporter dyes or stains. These things help us in recognizing whether the DNA stuck in that space or not. So naturally, we can use this for diagnosis for infections, and this can actually be very quick. It doesn't take much time to heat up DNA and cool it back down, but also for culturing of unknown bacterium or viruses. So if we don't know what it is, we can plug in what we suspect it is, and if it sticks, then it must be that. Whenever we talk about the size of DNA, it can actually tell us a lot. In general, the smaller the piece of DNA, the smaller the organism typically is. So like whenever we look at palindromic sequences, for instance, we're really looking at just a few base pairs, which is our basic unit. But as we look at bigger things, we see bigger numbers of base pairs. As a general rule, again, base pairs is the basic unit, which really represents a letter. So for the average gene in E. coli is around 1,300 base pairs or letters. But we can introduce things like the metric measurements like kilo and mega. And so the illustration is, again, we can say 1.3 kilo base pairs or 1,300 base pairs. PCR is the next important topic that we should cover. PCR is going to be a technique that was actually developed by a student initially. And what's cool is it's an ability to take and apply the normal processes of DNA replication, but in a unique way. So we can actually copy DNA outside of the host cell. And so the idea is we can make lots of copies of DNA that can allow us to do lots of different types of genetic work. So this is very helpful because it has, again, incredible specificity or sensitivity. As I just mentioned a minute ago, what's helpful is we're going to mirror the natural processes of DNA replication, but we're going to do so outside the cell. So in the normal setting, we would take and open the double helix with the enzyme helicase, but in PCR, we use heat instead. This allows for the DNA strands to be exposed and open just like they would be in our replication fork. But just like with anything else, in terms of copying DNA, we must first affix primers. So we still need primers. So naturally, the primers are where we get our specificity. If I design a primer to occupy a certain region of the DNA, that's all I'm going to be able to copy. But the last thing we need is DNA polymerase. That allows us to copy DNA from the primers on. So as we just discussed, primers allow us that specificity. If I point the primer at a specific place, I can only copy downstream from it. And so the idea is I get that specificity. But since we used heat to open up the DNA, we're going to have to use a special polymerase that is also heat stable. And so we actually stole it from a bacterium that's a thermophile. And so it's a unique DNA polymerase called Taq polymerase, which stands for Thermos aquaticus. And so the idea here is this polymerase works well at high heat, which allows us to, again, still use heat to separate, but also be able to copy DNA. So whenever we look through the steps, there's really three basic steps for this. We heat up to cause denaturation, we lower the temperature to allow for priming, and then we warm up just a little bit more to allow for extension. So whenever we actually look at this, note that we have our DNA that we're starting with. We denature by upping the temperature to around 94 degrees Celsius. We then have to cool the temperature, which will usually be the 60s degrees Celsius, around 65 degrees Celsius. Allows for the priming to occur, the addition of the primers. But in order to get Taq polymerase to work again, we need to use a higher temperature. So we got to crank it back up to around 72 degrees, which allows for that elongation or extension. So really 3 steps: heat up to separate, cool down to add the primers, and then warm up to allow for extension or copying. And so if we just keep alternating those 3 specific steps, we can end up with lots of DNA very quickly, because again, 2 strands can turn into 4 strands, 4 can turn into 8, 8 turns into 16. So this is that same basic doubling ideas we discussed with bacterial binary fission. So PCR is a super cool technique you should definitely know about. But there's a newer technique called real-time PCR, because with traditional PCR, you don't know how many strands of DNA you got until after you're done. But with real-time PCR, you can actually detect the amounts of product at any given time due to fluorescence. And so there's the idea that we can have real-time data in terms of the amount of copies we have. So what can PCR do for us? Well, we use it in everything like gene mapping or studying genetic defects in cancers, forensic studies, infectious disease, taxonomy. Literally anywhere that we're going to work with genetics, we're going to do PCR. And so PCR, incredibly important and definitely a topic you would want to know about. So really what this can mean is the opportunity to do anything we want genetically, including recombinant DNA technology, which is really about deliberately removing genetic material from one organism and combining it with that of a different organism. It's to create what we call a genetic clone, that genes from one organism can be physically moved to another one, creating a clone of those genes in that other organism. And so ultimately, since genes equal protein, we get the same protein product out that we would want, just in a different organism. But in order to move genes, we must be able to then isolate genes and use that gene product to make PCR copies. So the first thing we need to do is to take and bust open things like the cell or the nucleus, pull out and isolate the genetic material, and then use that genetic material to then make more copies of DNA. Which we can do through PCR. One of the things I want to point out is there are things like genomic libraries out there where they maintain very specific cloning hosts and vectors that allow us to create specific scenarios. So there are certain things out there that's easier to move genes in and out of, and so genomic libraries are really this culturing or continuation of these things so that you can easily have access to these resources. Cloning vectors are an interesting topic that we're going to spend some time on. Cloning vectors are very helpful in the movement of genes from one host to another. If you recall, vectors just move things, okay? And so what cloning vectors do is they move genes. And one of the more common cloning vectors we use is plasmids. Plasmids are, again, small extra pieces of DNA that are easy to manipulate. And as long as we can get the plasmid in a host, then we're good to go. Another strategy is the bacteriophage, or moving, again, genetic information using viruses. We're capable of creating a virus in terms of the genetic material in it and allowing it to infect other cells, putting the genetic material in it. So really two common cloning vectors, plasmids, and really viruses. So let's give you an example of a cloning vector. This is PUC19. PUC19 really has three major parts: the ORI, or origin of replication, the LAC-Z gene, and the AMP-R gene. So again, with origin, this is where we're going to start copying it. And naturally, we'd want to always be able to copy the plasmid so it can be passed on to future generations. So we never, ever, ever play in the origin. Do not put any genes in the origin. AMP-R is actually going to be ampicillin resistant. And so this gene allows this organism that takes this in to become resistant to the drug ampicillin. And so the idea here is we wouldn't want to play in this region either, because this is important for us to screen what has this information and what doesn't. So really where we play is in the LAC-Z gene. And what the LAC-Z gene does is take and break down basically a substrate into a product, but one of which the products is actually a blue color. And so what happens is if we insert PUC19 without disrupting the LAC-Z gene, we see colonies that turn blue. But if I take and insert DNA in the middle of this LAC-Z gene, will I still produce that blue pigmentation of the colonies? The answer is no. And so really PUC19 is all about putting genes into LAC-Z, allowing for the movement of genes into a new host. But again, if I see blue colonies, that means the genes I tried to move in did not actually get moved because the LAC-Z is still intact or in place. So we just talked about, again, an example of a cloning vector, but we haven't discussed a cloning host. The idea is a cloning host is something that should be able to be grown quickly, that isn't going to cause illness, that we know a lot about, that it readily accepts, again, information from hosts, and naturally we can replicate over time. So what could we produce through recombinant DNA technologies? Well, the answer is basically anything. If we need proteins, we need medications, we need enzymes, these are all features that can easily be grown through recombinant DNA technology. Naturally, things like immune treatments would be nice. The production of things like interferons, which are chemical messages to really fight things like viral infections and even cancers. Interleukins, which again, types of cytokines that regulate immune response, so used in cancer treatments. Tumor necrosis factor, which is used to treat cancer. The idea here is these products can allow us to naturally be able to fight viral infections and cancerous cells. Even hormones can be produced through these types of technologies. So for example, HGH, or human growth hormone, very, very important in the formation of appropriate development in life. And so we can use HGH to facilitate whenever it's deficient. Even enzymes can come out of this. So naturally, things like tissue plasminogen activating factor, or TPA, can dissolve potential blood clots. So enzymes and enzymatic activity are something that we can now help try to regulate. Or even the production of vaccines. Naturally, we can produce products that can be used to allow for vaccines to be developed. So whenever it comes to recombinant DNA technologies, the sky is really the limit in what we can ultimately do. As long as we're given an opportunity to experiment in this area, there will be huge applications that come out of this. But often, again, we need to move genes to other organisms, producing transgenic organisms. These are things that are going to have unique genetic modifications that allow them to produce specific products or have specific features. So we can use recombinant microbes and do things like bioremediation, where we fix certain issues in the environment by using microbes designed to carry out an action. So there are microbes out there that prevent ice crystals from forming, allowing for a lower temperature that doesn't allow for freezing. They can invade and destroy insects. They can be used to produce biofuels or even produce drugs. Really, with recombinant genetics, we can do almost anything. So there is some concern with all this in that by producing these special, unique transgenic organisms, we create plants that don't behave the way they should, or bacteria that are not natural. Again, that's understandable, but I want to point out that there is always genetic exchange amongst organisms. And typically those things that are genetically engineered are far more genetically stable and less likely to be passed to other things. So we have produced transgenic animals. As a general rule though, and whenever we mess with something's gene, its lifespan is typically much shorter. And so transgenic organisms become kind of risky in that the products usually don't have a long lifespan. So naturally we need to do more research in this area to allow for better potential outcomes. Unlike bacteria and yeast, animals do express human genes with very little issue, so we can make more very specific human products more easily in animals. And so that's a great potential future for us. Another reason why we like transgenic animals is the potential to study things like disease and genetic disorders in them, because again, they're animals just like us, and so there's more relatedness to it. They also have potential in really agriculture and pharmaceuticals. This idea that we can produce products and also still be producing normal agricultural product at the same time. So like this example of sheep and goats being basically turned into factories for pharmaceutical production, where we put genes in that allows for a specific product to come out, typically in their milk or semen. The idea here is we could harvest the drugs simply by milking the goat. Where this all tends to be a little more concerning is an area known as synthetic biology. And I illustrate the concern because this is the first illustration where we straight up produced something that is capable of reproducing itself. So the first time we created something that didn't exist before. This is the first time where we made a replicating cell that was nothing more than a bunch of chemicals. And so the idea here is by doing this, we can illustrate that we can make cells that are no longer broken, which allows us to replace damaged cells. That could mean we could fix basically anything. And so the idea of synthetic biology is slightly concerning because some people are afraid it's like playing God. Is there potential for things to go wrong? Yes. But if we do this in an intelligent way, the potential of saving lots of lives and fixing issues that have been to this point untreatable becomes a real possibility. So as I was just illustrating a second ago, there is ethical concern with this. Fears of creating something that we can't control is always a real possibility, but without experimentations, we don't get the advances. Again, we need to experiment to understand what potentially we can do. And so it's important to understand that science can't move forward without experimentation. And so we really have to consider, what are we willing to risk? Is it okay for us to try to design cells that now replace damaged nerve cells? So now a person that has issues with their nervous system, like say a severed spine, can now walk again. Or do we consider that a bridge too far? And so the idea here is, without experimentation, we can't make the advances. We must be able to experiment, but within reason that the society is okay with what science is doing. So one of the first things that is a real possibility, again, this stuff really not that testable. This is more of an opportunity to illustrate the potential. So one of the things that is a real potential in the future is gene therapy, where there are several diseases out there where literally a single gene is broken. And as a result, there's a physical illness that is for the rest of their life. So the illustration of type 1 diabetes is really caused by lack of insulin. So if we could supply the ability to produce insulin again, we can fix this issue with no other need for recombinant technology in terms of harvesting insulin from fungi or grinding up pancreas of pigs and cows or any of that anymore. The idea that we can physically fix a person so that they can produce their own product is what gene therapy is about. The reason we care so much about gene therapy is this is the potential to correct or repair literally a problem that they're stuck with for life. And so this is a permanent fix for the issue that they have. And so having this ability would be incredibly helpful to fix a lot of people and help save their lives. So one of the things that we thought about doing was to basically take the normal gene cloned into retroviruses or adenoviruses and have them basically be able to insert genetic material into the tissues. And so those cells can then be reintroduced to the patient's body and would hopefully then replace the cells, now producing the appropriate outcome. So right now, I want to point out experimentation and clinical trials of gene testing is still very, very, very new. This is an area that we haven't got a lot of opportunities to experiment with, but this potential is so amazing that we could save lots of lives. So trying this out on literal humans is very important to the process. Right now, this is a volunteer basis, and specifically, these usually are genetic defects that we're looking at, and currently most of the research is going on in cancer, and so naturally we could try to even fix cancerous cells so that they're no longer cancerous, and so the idea is we're trying to again work in this area, but there are lots of safety concerns, specifically about like the delivery of the resources by primarily viruses. But what's more realistic in really our lifetime as being commonly used is something called germline gene therapy. So rather than going in and trying to fix all of your cells in your body that are broken, let's fix a sperm or egg, or at least a very early embryo before it's divided a bunch. It's much easier to fix one cell or a couple cells versus all the cells inside of you. And so with germline gene therapy, we can literally fix the problem before the problem becomes an issue whenever it turns into a person. So what could we do with this? Well, ultimately we could get rid of things like Alzheimer's. We can prime the body in such a way that viral disease becomes no more of an issue. Same with cancer. And ultimately, again, even controlling what genes are expressed and not would be a very helpful proposition. Naturally, we can also use this for medical purposes. One of the things that we're looking at is the microRNAs or miRNAs that are used to actually stop genes from being expressed. So if you have a broken gene, let's turn off the gene so that it doesn't produce the outcome. So again, if we turn off the broken cancer genes, hopefully we wouldn't see cancer form. And so the idea is we can even use microRNAs as just a therapy to help fix the current problem. Ultimately, this inhibiting factor again stops really the progression of disease. So if we use it to say maybe work with cancer, we physically stop the cancer from progressing, which is a wonderful opportunity to allow us to give us the time to really fix the real problem, which is the cancer cells. Inhibiting a microRNA in African green monkeys lowered their plasma cholesterol levels. So now we're literally talking about controlling cholesterol with microRNAs, or even again certain what's considered to be lifelong viral infections, so like hepatitis. MicroRNAs, even as a simple therapy, could be incredibly helpful for us. And really, we've been using them with a great or high success rate. We've seen the first examples of a nasal spray stopping the spread of RSV. So really what we saw was the microRNAs basically decrease the likelihood of a highly transmissible virus. And so the idea here is we can literally help stop or at least half a lot of infections that occur out there. So the next thing I want to talk about is really how we can analyze or understand DNA, which includes genome mapping and profiles. The idea here is by looking at the genes, we can learn a lot about people. And this does, again, raise that ethical issue. What's okay for people to know about you based off of your genetics? There's a real possibility that we can literally tell people whether or not they're going to get cancer or Alzheimer's or other very problematic diseases that would develop usually later in life. And so some people wouldn't want to know this information, nor would they want other people to know this, like, say, their employer, or again, the healthcare provider that they're going to be utilizing for insurance later. And so again, these are things that get kind of touchy. But really, sequence maps tell us the information we need to do all the work we need to do. And so we need to know the sequence before we can manipulate the sequence. So there's lots of sequence maps out there. There's published genomes for huge amounts of viruses and bacteria, and even some eukaryotes. They tend to be smaller, simpler eukaryotes, but they're also genomes of eukaryotes. So how can we learn these sequences quickly? Well, that's where the whole genome shotgun sequencing works. If I was trying to read all the letters from start to finish, that would take a lot of time. But if I cut it in a bunch of little chunks and read all the chunks at the same time, I can get it done much quicker. And so the idea is the fragments can be basically read much quicker than the whole genome itself. And so by breaking it down, we can get through it much more readily. Ultimately, a computer is going to do all this kind of stuff to allow us, again, to quickly get the information. And this is really where we're seeing a lot of current research, where we're just trying to sequence literally everything on the planet. By having all the information, we can then use the information to fix issues or know what things can be passed from one organism to another even. High throughput sequencing is another system. Again, what's characteristic of this is it's a sophisticated computer system that could literally, again, speed up the whole process here. Again, high throughput illustrates that we can rely more on the machines to get these materials done. And so I'm not going to test you on this, just understanding that we are developing greater techniques every day to be able to sequence and understand what genetic information is out there. And this has really led to genomics and bioinformatics. So genomics is, again, the understanding of what genes are out there. And then bioinformatics is, again, the info to know what we can do with it. So one of the things that would be helpful is, again, having all the information of what's good or normal and what's, again, not normal. And that can tell us a lot, like how does disease develop or how aging even works. And so going through and looking at normal versus non-normal, we can really help try to, at a genetic level, understand disease. DNA profiles was something that I was kind of illustrating or hinting at earlier with this idea that this is maybe information that people don't want out there. But I want to point out that DNA fingerprinting or typing is a common strategy that's existed since the 1980s. And it's been used in lots of different ways for both good and potentially bad. Crime scene investigation is the easy example where people, again, can routinely see how it can be used. It's not uncommon for individuals that go into the prison system repeatedly to have a DNA sequence taken and be stored on file. So if they commit crimes in the future, it's quickly tied back to them. And so naturally, again, through things like the gel electrophoresis and RFLPs, we can again see comparisons of genetic material that would tell us what's going on. So notice here we have the victim's DNA, we have the evidence 1 and 2, again illustrating same genetic evidence line for line, and then we have suspect 1 and suspect 2. If you note, suspect 1 matches identically to the evidence, which suggests that their DNA was the one found in the crime scene. And so the idea is this is literal proof that the individual was involved in some way in terms of having their genetic material at the scene. And so again, those RFLPs or restriction fragment link polymorphisms, that's how we do this. If we cut, again, a blunt cut, we can create chunks of DNA that can be passed through a gel to create banding patterns that give us information. Each of these samples, again, would typically be digested twice over with 2 different enzymes, because again, there is a slight chance that you can use one enzyme and get the same banding pattern, even though it's not the same DNA. But if you do it twice with 2 different enzymes and you get the same exact banding patterns in both categories, that should show absolute identity. So the next thing I want to talk about are single nucleotide polymorphisms, or SNPs. These are important changes in a genetic code that allows for a specific trait to exist. And so the idea is, by studying SNPs, or single nucleotide polymorphisms, we can learn a lot about how changes happen genetically, and basically allow us for an understanding of how diseases can develop. So notice here we had a single nucleotide change that allowed for the production of an alternate protein, or no protein at all. And so the idea is single genetic changes, or nucleotide polymorphisms, are enough to create disease. So by understanding SNPs, we can understand disease. Microarrays are the next big step in that we're going to combine biology with computer science. And so this is really the ability to track gene expression by using basically markers. And we can actually have a computer do this. And so we can literally track the expression of thousands of genes at one given time. So what could we use this for? Well, honestly, some very sensitive and effective diagnostic tests. The idea to not only type cancer, but even subtype it very quickly gives us lots of opportunity to help save lives. The last real slide here is on proteomics. And I know I kind of mentioned it previously, but proteomics is the future. This is the study of proteins, how proteins are made, and what proteins do for us. Ultimately, proteins are still the bigger topic here because they result in the physical outcome. So the study of proteins, or proteomics, is actually very much the future. And I encourage anybody that's truly into this stuff to take a look at proteomics, because that is where the science will advance next.